thanks everyone for the opportunity to present some of our ongoing work with you today. And so today I'm going to focus on some areas in my lab that we've been focusing on at the interface of nanomaterials, in particular nanobiomaterials and biological fluids, and how we try and modulate this interface to better enhance immunotherapy and targeting of cells in general. So shown here, the slide progresses. Here we go. Shown here is how I break down my lab into three different areas. I'm an immunoengineering lab, similar to Daryl. I have not been around as long as Daryl, who essentially started this field. But my background is mainly in biomaterial science. And so we still have a huge focus on developing different types of hydrogels and self-assembled systems. We still do a bit of polymer chemistry in my group. And we try to understand how these materials interact with proteins in biological fluids, and then how these interactions and structures of the nanomaterials influence the immune system. So we do quite a bit of basic immunobiology. Biomimicry is involved in there. How do we mimic some of these immunological interactions that go on that control immune responses, and how do we modulate specific cell subsets, so specific types of immune cells, to better control inflammatory immunosuppressive functions. So we take what we learn from there and we try to apply that to actual therapeutic applications. And these are some of the areas that we work in, shown over here. At first, it seems like a lot, but in the end, it's actually, as I'll show in the beginning of the talk, it's really applying a similar delivery system, but being customized for different applications. And I'll show why we can work in so many different areas just by customizing the same or just a few different delivery platforms. And one thing I want to point out here is that we do a lot of theranostic work. We found that for targeting cells in the immune system, it's much better for reproducibility to be able to trace what immune cells we're modulating. It's just much better for us to trace, the network of immune cells that have been modified in different cases for reproducibility. So theranostic is a big area of interest for us. So everyone here is likely familiar with nanomaterials, of course, but I just wanted to clarify a few things because there's so many terms being used and thrown around these days, like nanomaterials, nanoparticles, nanocarriers, nanodrugs. And I like to use nanobiomaterials and nanocarriers. And I just like this distinction with biomaterials just because there are a lot of nanomaterial platforms that just aren't ideal for biological applications. So I like to use nanobiomaterials, but fairly interchangeable with many of these. Shown down here are some of the more common platforms everyone here is, I'm sure, familiar with. I just want to point out, I don't really work with any of these platforms. We work with self-assembled polymeric systems that take on different structures than shown here. We do work with micelles, and we do work with polymersomes, vesicular structures composed of polymers similar to liposomes, but we actually don't use any of these classical lipid-based or gold or quantum dot-based delivery platforms. What I'm I don't want to say happy about, but very glad to see nanomaterials getting some attention in the media these days because of the recent usage and requirement for mRNA delivery. It's really brought to the forefront how these engineered systems can be customized for delivery of specific molecules and drugs that otherwise would be very difficult to control and deliver on their own. And so these lipid nanoparticle systems are actually quite old, but very interesting systems that I'm very glad to see being applied in actual clinical setting currently. And the interface here between these materials is pretty critical. We have right here this PEG coating, which I'll talk a little bit about, which has also been thought about in terms of being a source of allergy and response during some of the side effects for using these vaccines. To clarify, again, a lot of you are probably familiar with these techniques. These are just the main techniques that we use to characterize our materials. So some of the data I'm going to be showing is going to be using these techniques. Dynamic light scattering and nanosite shown here in the middle, where we can actually see the particles and look and calculate their diffusion rates based off of their actual visible motion. We do a lot of X-ray scattering, small angle X-ray scattering at Argonne National Lab. And we do a ton of cryogenic electron microscopy. It's really the best way for us to see the structure of our materials in aqueous environments because we work with self-assembled materials. And this is just a good example of the biomimicry we attempt to do in our lab. At the top here are filamentous and vesicular nanostructures, kind of an elongated vesicle here. We try to match the structure. This is influenza virions down here in both a filamentous and a vesicular form. And This biomimicry comes mainly from this slide here, which is that nanomaterials and nanobiomaterials are actually quite natural. They're found throughout the body. They participate in a variety of different applications and signaling pathways. Tumors, of course, release exosomes. Neurons can communicate via extracellular vesicles. Of course, we have lipid transportation through HCl particles, which is, of course, huge. And then we have, for us, which is really interesting for us, is this communication that happens between immune cells that involves exosomes, as well as uptake of nanoparticles. So the ability of APCs, antigen-presenting cells, these are scavenger cells of the immune system, I'm sure you're familiar with. Their ability to take up small particulates is really critical for mounting immune responses against viruses and bacteria. So we really want to hijack and take control of this kind of process in order to communicate with very specific and selective immune cell populations. And how do we go about this biomimicry? We like to use the virus as our model system, which has a size range about 20 to 100, sometimes up to 200 nanometers in diameter. And we can control the size, the shape, the charge, and the surface chemistry of our materials. We engineer these into the particles to control how they interact with cells, how they undergo biodistribution throughout the body, and how they can target selective locations. But many of the strategies that we use are taken directly from viruses, and I'll show a few examples of how we do that. A few examples is, of course, being able to have different ligands displayed in organized fashion on the surface of the particles, the size range, and the ability, of course, to have hydrophilic payloads loaded inside of the interior for enhanced intracellular delivery. This all comes down to the bigger field of ours, which is controlled delivery, and controlled delivery specifically in the immune system. So, of course, everyone's familiar here with cancer immunotherapy and how this was such a big deal. This was the breakthrough of the year in 2013. And back then, and continuing to this day, we still realize there's a lot of side effects that can occur with these different kinds of strategies. And so being able to control which cell types and particularly which organs and locations these particles are going to modulate the immune system is really critical. And drug delivery has progressively become a bigger and bigger part of immunotherapy. Shown here are just a couple examples. This is a paper from a few years ago looking at slow release hydrogel systems for immunotherapeutic delivery. And of course, there's the classical system here where you can actually put all kinds of different cargoes, like mRNA I was discussing before, inside of a nanoparticulate and control their surface chemistry in order to control where they go and which cells they are actually going to be targeting. So for us, again, the main goal is to target different cells and organs of the immune system. And so we target quite a bit the spleen and lymph nodes, depending on the application, also the lung in our tuberculosis work. And many times also important is how do we avoid uptake by certain cell types? And that goes into the spleen as well as the liver, depending on the application. You want to avoid the immune cells in those locations. And for the vast majority of interactions, we're focusing on these APCs, these antigen-presenting cell populations, dendritic cells, B cells, and various other myeloid cell populations that take up particulates, process them, and present them to cells in the immune system. So we see these dendritic cells as a central node within the immune system. As Daryl mentioned, by targeting these cells, we can actually go on different routes of the immune response. We can go pro-inflammatory or we can go anti-inflammatory. In this case, I'm showing tolerogenic. And so briefly shown here is how that works. So this is the same cell type, but depending on what you send to the cell, as well as the chemistry of what you send to the cell. So and this is where the engineering of the nanoparticles and the polymer chemistry comes in, is if you can design materials that can carry certain payloads, interact with certain cell receptors, like these toll-like receptors, both inside and outside of these cells, as well as respond to intracellular chemistries, like in the endosomal compartments to cause controlled release, sustained release, all kinds of different times of delivery within inside of the cell, as well as escape from these compartments to reach the cytosol. And this is all going to be influencing cell surface receptor expression. So there's certain receptors that express on the cell, costimulatory molecules that go to a pro-inflammatory state, as well as target molecules like peptides that complex on the surface of the cell that become the targets of the immune response. And the third arm of this is controlling the cytokine. So how these cells are activated changes their hormones, their immune hormones that they're released, form of cytokines, all to activate different types and subsets of these effector T cells. And this, of course, really critical for pro-inflammatory pathways like cancer immunotherapy and vaccination. Now, on the other side, if you deliver something as anti-inflammatory that targets different receptors, goes through the same intracellular pathways, but results in activation of different cytokine expression, so different combination of these cytokines, and decrease or inhibit those co-receptor molecules. So we want to inhibit those, and we call it, it's a blockade essentially. So I'll give an example of how we can deliver to result in a costimulation blockade is what that's called. But we still want to present antigens on the surface, and we still want to activate T cells, just different types of T cells, regulatory T cells. So by engineering our materials and loading different payloads into them, we can go through either of these pathways with dendritic cells. And not just dendritic cells, but other myeloid cell populations, but the DCs, of course, are the major cell type of interest here. And what do we want to deliver? It's really these two categories. An antigen, which is some kind of target molecule, in this case, usually a peptide, but I'll give an example where a lipid can also serve this purpose, but in some kind of immunomodulator. So the target for the immune system, as well as the signal direction you want the immune system to go in. So in the course of an vaccine response, you have an adjuvant, a pro-inflammatory response is the immunomodulator, is your result you want to get. Whereas in tolerance, something like vitamin D, something anti-inflammatory would be the immunomodulator. And I'll give an example of each one of these different kinds of pathogens where we've either modulated them, mimicked them, or taken some kind of biochemical component to actually incorporate into our delivery system to elicit a controlled immune response. So that's really the strategy that we're using. I just wanted to briefly go over like how we want to interact with the immune system and the direction we want to go in with our materials. So now I want to go and go to transition to the types of materials we're actually using. So what are the biomaterials and the delivery systems that we work with? And again, those are nano biomaterials primarily. But to start off, I want to go over a couple things really quick. First is about the press that nano biomaterials have been getting in the last, I would say, 5 years or so. It's come to the forefront that, the targeting doesn't actually work as well as we had hoped. A big paper came out, several papers have come out discussing this, particularly from Warren Chan's group, discussing how if you're targeting a tumor, 90% of your payload in your nanoparticle is not actually going to get to the tumor. Only about 3% in most cases is actually going to get there. We still have issues with overly complex chemistries that are difficult to scale up for actual clinical usage. And there's still loading difficulties. Many times we still have to customize the delivery system for the actual payload. So if you're delivering DNA, you have to have something customized for loading negatively charged material. So you have a cationic type of polymer or lipid being used. And so that presents difficulties when loading multiple types of payloads that differ in their chemical, physical chemical properties. So for example, if you want to load a vaccine that has mRNA and a small hydrophobic molecule, and something that's charged, that's positively charged as well, it's difficult to make a single nanocarrier that can compensate for all those different properties. So multi-drug loading is still an issue in many cases. But with that negative side being said or put forward, there's a ton of benefits still to using nanomaterials, and this is still, I think, booming and a great area to be in, especially with the increased publicity from these successful vaccines currently. Critically, we have much better targeting. Even though we have off-site effects, we still have enhanced targeting of very specific locations in cells. This can result in decreased drug dosages, decreased side effects and toxicity. One thing that we've recently got into is controllably repurposing drugs. So if there's a certain pathway that you want to tap into or pathway you want to avoid, you can engineer your nanomaterials to control which cells the drug goes to better control the type of therapeutic response that you get. So I'll give a few examples of repurposing drugs. And many times you can completely change the therapeutic effect of a drug by controlling where they're actually going in vivo. And finally, I already mentioned the advantage is being able to trace where you're actually targeting, where your drugs are going using a theranostic application. And as I hoped I introduced in the beginning, I believe that nanomaterials are ideally suited for targeting the immune system. There are, of course, many applications where maybe nanomaterials aren't the best. You don't want to use the same tool for every application. But for the immune system, I really believe that because of this evolutionarily driven interaction between nanoparticulates in innate and inflammatory immune cells, this is an ideal platform for modulating the immune system and communicating with these cells. So here's the systems that we work with. We work primarily with self-assembled blockopolymers. The main one shown here is the one I'm going to focus on today. Is polyethylene glycol as the hydrophilic site and polypropylene sulfide as the hydrophobic domain. And by controlling the ratios of these two different blocks, we can control the volumetric ratio and curvature of the different particles that assemble. And so we can use the same overall chemistry. Just by changing the block lengths, we can actually specify different self-assembled structures. And so here are the main structures we work with: micelles, phylomicelles, polymersome vesicles, and the bicontinuous nanosphere. There aren't many groups that work with this structure. I think we were the only ones to actually try using these in a therapeutic application. But these are very similar to cubosomes. They're polymer. So cubosomes are formed of lipids. These are, say, cubosomes formed from polymers, and we can control their internal structure quite a bit, as well as their overall structure. So we can make them very nicely spherical for controlled delivery applications. And this is, of course, fundamental to biology, the self-assembled system. I don't have to explain this to this crowd. But I do want to point out that although this is our main system, we do look into other systems, particularly with a lot of focus being placed on PEG not being always the ideal hydrophilic polymer due to allergic reactions and things that are popping up due to our overuse, not overuse, but consistent use of PEG through many different applications. In society, people are exposed to a ton of PEG. So we have a lot of high throughput systems for customizing, testing, and just looking at, what kind of structures are going to be assembled from our new chemistries that we're making. So every time you change one of these blocks, you change its chemistry, you have to, again, reassess how it's going to assemble, because the solubility properties, the physical chemical properties of the new structure is really going to dictate, the structure that's going to be formed, the nanoparticle that can form in the end. So we have a lot of high throughput methods for assessing what kind of structures we're going to get from new polymers. So again, I'm going to be talking about today just one. It's really one system, but it has two parts. First is the block copolymer system, the PEG PPS system, polypropylene sulfide linked to PEG. But we recently have a new system, ppsu, which is polypropylene sulfone. It's the oxidized form of polypropylene sulfide. And the reason we started working with this is that the sulfone is actually the final oxidized form of our polymer when it degrades. So when this drug or this molecule is delivered in vivo, it oxidizes slowly over time. And this hydrophobic domain, this is one of the usefulnesses of the PEG PPS system, becomes hydrophilic. So it actually is the release mechanism. So once this becomes hydrophilic, you're disrupting that ratio of the hydrophobic and hydrophilic blocks. You can actually have structures reassemble, and you can have the payload be released because the particle now falls apart due to the oxidation shown here. So I actually wanted to have a student make a completely PPS-based system that had oxidized PPS on one block and the hydrophobic PPS, polypropylene sulfide, as the other block. And I thought this would be a nice way to control the system and make a new kind of block that we can make everything in-house. We don't have to buy our PEG chains anymore. It turned out when he made this polymer, it actually was not water soluble like we thought it would be. And so for some reason, the polypropylene sulfone system was aggregating. And we went through a lot of work to figure out why that was occurring, and it ended up giving us a new delivery system. So the two systems we're going to talk about today are our block copolymers, PEG PPS, and a homopolymer. So this is not a block copolymer. It's a homopolymer that actually self-assembles in a controlled fashion on its own, which we think is a nice new platform for controlled delivery. And shown here is just some of our publications and showing that we use all these different structures for different applications. I wish I had more time to talk about the BCNs because I really like this system, but for the sake of this lecture today, I don't have time to go through them. But if you have questions about the BCNs, I'd be happy to answer them at the end. Here are some of the advantages of using the PEG PPS system that we've found over the years. One is that we can scalably fabricate all of these structures. Many times it's difficult to assemble uniformly different types of nanostructure assemblies or morphologies. We can actually use flash nanoprecipitation to actually form all of our structures in a high throughput manner. So we can make gram scale amounts of these nanoparticles fairly uniformly, very quickly. Drug loading efficiency is something I'll talk quite a bit about today, which is an advantage of the PPSU system. This is the theranostics I've already talked about, where we can trace the organs that these cells are going to, the organs and cells that these particles are going to. And importantly, we have done some non-human primate studies showing that the biodistributions we see in mice do carry over to higher order mammals like primates as well. So we're very excited about that. Intracellular delivery is an advantage of our system. As these particles degrade, they act as small surfactants that insert inside the membranes of endosomal compartments, allowing a burst effect. So it's a temporary release of a payload into the cytosol. So it actually is able to release into the cytosol without disrupting the endosomal compartment. So many other methods like the proton sponge effect, which actually isn't completely verified, can result in complete disruption of the endosome, and which can be toxic. So our systems don't rely on that, and they can have very safe release of payloads into the cytosol. One thing I want to point out about the PEG-PPS system is that it has a very, very low immunogenic background. So many polymer systems actually have a background inflammatory signal. So no matter what you do, there's some kind of response to the polymer system. That could be for many reasons. And PLGA, for example, is one example that many times is used in an anti-inflammatory fashion. And so it has a background in immunostimulation, mostly anti-inflammatory. And if you compare that to the PEG-PPS system, the levels are much, much lower in terms of the amount of backgrounds that you get. And so for us, we like that because we can better customize the payloads to get a certain controlled response, as opposed to having to worry about what is the polymer going to do in terms of the immune system as well. And we still see this nice thing that carries over from other nanoparticle systems where we can decrease side effects and toxicity and dosage when delivering things with our PEG-PPS systems. So I want to talk about today, for the sake of time, just two of our newer platforms that we're working with to kind of give examples of how this nano biointerface was important, as well as biomimicry was important for us. So shown here is our sustained delivery system that we developed to slowly release nanoparticles in vivo. We were trying to mimic the fact that you can actually make, as I mentioned before, our bodies actually make nanoparticles all the time. Viruses are budding from membranes of cells in the constant rates, or can do this in the regular rates. And so we wanted to see, can we actually have a reassembly in a controlled fashion to make nanoparticles in vivo? And this was what we had as an idea. Many of you are probably familiar with this, but there's because filaments and phyllomicelles many times are in a kinetically trapped state, they will slowly over time convert to a more thermodynamically stable conformation, usually the spherical structure of a micelle. So we see this without filaments. If they're sitting in storage for about a month, they start to slowly transition into micelles, and we would see this using electron microscopy. You'll see this always happening, budding from the ends. We've done cryogenic tomography, cryo-EM tomography to see this. We had a reviewer actually ask us, are we actually looking at filaments coming head on as opposed to actually micelles coming out? So we had to do this in 3 dimensions. And this took a little bit of time. It was kind of expensive. So I only have a couple of these pictures. But this is verified that we were actually having this transition occur. At the ends, you can see this happening. And this has been known to happen with filaments and other systems as well. Our system, I'll talk about this, but it's dependent on oxidation. So we can control this quite a bit. But we can take these filaments and we can cross-link them together to form scaffolds. And to do this, we actually incorporate a cross-linking group. What I like about self-assembled systems is that we can mix different block copolymers together to get different functionality. In this case, we can mix a standard block copolymer, PEG-PPS, with a functionalized version to control the amount and spacing of cross-linker groups inside of the filament. And we can also put, of course, drugs inside. So here's an ethyl eosin payload with inside of a filament. Cross-link this together. It can form in situ, so you can have a liquid, you inject the liquid, it'll form a gel in situ really quickly within a matter of seconds. And these are fairly stable. The vinyl sulfone actually interacts with the tissue to hold it in place really nicely. And so we haven't had any issues with inflammation with these in mice for quite a bit of time. This is the synthesis that we use to make the vinyl sulfone. We had to make a heterobifunctional version of our block copolymer for this. It took a little bit of optimization, but this has been shown before, so it wasn't too difficult to do. But this is the block that we're mixing in at different amounts to control the cross-linking within inside of our filament system here, this vinyl sulfone functionalized PEG that we attach to the PPS group. And so in doing this and making these hydrogels that undergo this filament to micelle transition, we actually believe we actually have a different mechanism of hydrogel degradation. So it's shown here the 2 different classical methods, the surface erosion method, where you have at the molecular level, you have degradation of the polymer given off monomer over time or some degradation product, a small molecule degradation product, and it eats away from the surface inward. You have bulk degradation where you have blocks and it can be breaking apart throughout the entire hydrogel, and you have irregularly shaped components or pieces breaking off. In our system, we only have highly monodispersed micelles being given off as a degradation product. So in this case, this is a mold we use, like just a shape. We can of course shape these by cross-linking them in various molds. This is just an ex vivo made hydrogel that we can trap various payloads inside. We can trap cells inside. It's very biocompatible. But when it degrades, regardless of the cross-linking density, because it's actually the ends of the filaments that are switching over to these micelles, we have pretty much only uniform micelles being released as the degradation product, which we use as a delivery platform. So you start out with these gels, load proteins or loads molecules or therapeutics inside of the gel, and it will slowly release over time as these micelles are released. It could be either freeform or trapped inside the micelles. So how is this working? We did a lot of modeling, primarily with Ken who's a material. Main sources of the free energy you have to compensate for. You have steric stabilization, of course, of the peg chains on the outside. You have chain stretching of the hydrophobic content on the interior, and you have the interfacial tension between the hydrophobic domain and the solvent on the outside. And so you balance the free energy of the system. You can figure out what is the optimal equilibrium radius of these particles for the cylindrical as well as for the spherical shapes. And we see here, this is the sphere, and we see that the cylinder has a smaller internal radius, resulting in less chain stretching. So it's actually the preferred structure at higher interfacial tensions. And so this is what we saw with the modeling as well. What is the lowest free energy state for these different equilibrium radii? And so shown here at higher interfacial tensions, the cylinder is preferred. But as you decrease interfacial tension, there's a transition here where actually the sphere, which can now compensate and have a larger radius due to the fact that you can have larger amounts of chain stretching occurring and stabilization of the outer peg corona. And so once you get to this transition point, it actually turns out that the sphere actually had the lower energy states for its equilibrium structure. And so we can actually induce this by modifying the interfacial tension by actually oxidizing our polymer. So this hydrophobic block, once it becomes more hydrophilic, the interfacial tension is going to decrease. And so we did a lot of droplets. This is a drop shape analysis looking at a chloroform water interface with our block of polymer. And you can see that as we oxidize, this is done with hydrogen peroxide, you actually see a drastic decrease in interfacial tension as the PEG PPS system oxidizes. So what we're actually seeing is that as the polymer oxidizes, it actually promotes the transition to the micellar state. And so we can control it, the rate of the actual micelle production based off the oxidation of the polymers. If we can trigger that in some way, and this actually happens naturally in biological fluids or upon exposure to air as well, we can control that. So shown here is one system that we worked with. I have to say this is kind of a overdone system. We did this because it was a higher impact publication, but we typically wouldn't have so many different block of polymers thrown in together. We just wanted to demonstrate the capability of the system. So we have two imaging modalities. We have the crosslinker, we have the main structural polymer, and we have a payload, a model payload, all thrown inside to make this filament here, which has controllable cross-linking density based on the amount of vinyl sulfone block we put in. Inject these in the mice, form hydrogels, and we want to see how long will this release, how long will this stay. We've actually shown they can stay and release nanoparticles inside of mice for up to about 2 months. Currently, and we could potentially change that a bit more by adjusting the cross-linking density. And a really critical question we wanted to answer was that as these degrade, do they actually carry the payload with them? So does the payload that's loaded inside the filament actually transition into the micelle when it's being released? So we did a lot of work looking at this, essentially looking at the downstream effect. Like, what are we seeing inside of cells? Do we see the same ratio of the fluorescently tagged polymer as we do as the fluorescent payload? And the ratio of the fluorescence of those stays the same, regardless of the cell type that we're looking at. There could be more or less of each there, but the ratio stays the same, depending on the amount that the cell took up. So we are fairly confident that the payload is staying with the polymer as it's being released from the delivery system. Without that, they would separate, and you would see two different signals, some cells taking up one fluorescence, the other cell taking up the other. And really important for us is that we can sustain the modification and uptake of these polymers by immune cells for that 2-month period. So shown here is at the end of that time point. if you were to say inject these polymers sub-Q, would you see dendritic cells or macrophages or myeloid cells positive for that payload after 2 months? And the answer was no for most cases. But in the slow release system, yes, we can still have a good amount of presence of these particles still inside of immune cells at that later time point. So we see this as a nice way for slow release for many different applications, for slow release of anti-inflammatories. It could be for controlling the kinetics of a vaccine being delivered. We've also looked into this for cancer. It turns out we can actually hijack and deliver nanoparticles to modulate draining lymph nodes from a tumor site. So say, for example, you want to have so a tumor is, of course, draining its antigen to draining lymph nodes at all times. Can you actually modify the response that the immune system is having to those, which is typically tolerized in the case of a tumor? Can we inject sub-Q these particles or these hydrogels and over long periods of time modulate the immune response in those draining lymph nodes? And so we have found that we can actually target immune cells in the draining lymph node as well as actually have uptake inside of the tumor. Many of these myeloid populations, MDSCs actually turn out to be taking up these micelles quite nicely if they're peritumoral, if a peritumoral injection is used. So that's our slow release system that takes, of course, advantage of the interface, the interfacial tension that occurs between our self-assembled systems and water. And I want to briefly talk about our newest system, ppsu, which is, of course, again, a homopolymer that we just published, demonstrating that we can have self-assembly from a simple homopolymer block. It doesn't require PEG. And this is all due, again, to solvent interactions with these polymers, so the interface of the polymer and the solvent. In this case, we're using solvent polarity to induce an understudied form of electrostatic interaction, which is between sulfones. So this is the end product of oxidized PPS, is the sulfone group here. And so we, as I mentioned before, I wanted to make polymers of this for controlled self-assembly. It turned out these aggregated for some unknown reason. So we did a lot of modeling with Monica de la Cruz. She did a ton of atomistic modeling. This is her area. I don't do that, but she's probably one of the top in the world in that. So we worked with her and we have a nice collaboration going. She did a ton of atomistic models, and now we're shifting to coarse grain modeling to look at the more macro scale effects that we're seeing. But essentially, you put this in a perfect solvent, which in this case is DMSO. You can just look at the structure of this and say, okay, yes, this is something that would be nice in DMSO. And you see a fully extended chain of the polymer. So it fully extends, is very hydrated, very well hydrated, solubilized in DMSO. If you actually change slowly to a water as water as a solvent, so higher polarity solvent, it starts to aggregate together. And we believe this is due to screening effects. So initially you have a lot of repulsion between these oxygens shown here. And higher polarity solvents, some of those effects are screened slightly, allowing electrostatic interaction between a dipole that exists between the oxygen and the sulfur here. So it's kind of like a zipper effect. And at lower concentrations, what we found was really interesting is that the polymer folds on itself. So the modeling actually revealed this to us. The polymer actually has a propensity to form zippers with itself. And so the ability for it to form these zippers is dependent on the persistence length of the polymer chain. So it'll fold until it can no longer bend any further. And so what we end up with is this really interesting supramolecular amphiphile. So we have a aggregated crystalline interior, but we have these coils at the end that are, sorry, my mouse isn't moving. These coils, sorry, there we go. These coils at the end are actually water soluble. So the ppsu is water soluble. It just undergoes this zipper effect to cause it to aggregate. But if it's too short of a chain, it will no longer aggregate with itself, and this chain would stay at the end and likely form a kind of a hydrophilic component of a block. So this is a supramolecular amphiphile, essentially. And so these would then self-assemble into a variety of different nanostructures. And so we've tested this, and what's interesting is what we're calling the hydration history is what's really important here, is the rate at which you add water to the system and the amount of water you add to the system controls the kinetics of the system. So it's dynamic, essentially. So shown here is a good example. If you have 25 mg per mil of ppsu and DMSO, and you add water to it, either in this case here, 40 additions of half a mil, we form these filamentous, like nanostar kind of structures. If you add the same volume of water in 2 additions, 10 ml each, we get really nice vesicles. And if you add it all in one lump sum, all of the water at the same time, we now get a solid core micellar structure. And so this is where the Sachs analysis comes in. we are fairly sure that we have these different kinds of structures based off of the X-ray scattering analysis, as well as the electron microscopy. You can see these are vesicles shown here as well. So simply by taking this, it turns out to be a very simple and very rapid way of making nanoparticles. But the very best part of this actually was the fact that it turned out we could load any payload into these, regardless of the chemistry of the molecule, very easily at very high efficiency, greater than 90% for almost every drug we've thrown at it or molecule we've thrown at it. And so what the modeling with Monica has revealed to us is that we actually have a network forming in solution of these supermolecular amphiphiles. And we know this also physically because we can actually form gels under certain conditions. This will form a gel under certain hydration rates. And as we add the water to the system, it actually starts to compress. The network compresses as you have more and more zipper formation occurring between supermolecular structures. It compresses down into these nanoparticulates, and which was essentially, we believe, collecting essentially any particulates, any drugs that you have in solution. So regardless of the chemistry of the molecule that's present, if it's in the DMSO or if it's in the water, it will be collected and compressed into these particles. And we did FRET analysis to assess this as well. So we have 2 FRET pairs in solution. You have low signal or no signal at all. We do this process of adding water to the DMSO solutions, and you actually see the FRET effect occurring as 95% or more of the FRET pairs actually become compressed inside of the particles. And we can load, we've loaded almost everything we've thrown at it into these particles. This is just an example of loading fluorescent dextran into our particles. You can precipitate these down, do centrifugation, and get really nice particles. And really important is that it doesn't matter if you have different combinations of these particles, of these molecules as well. So we're actually investigating this for multi-drug encapsulation quite a bit for vaccine optimization. So if you imagine a vaccine, many times you want to have many different kinds of nanostructures, many different kinds of payloads in there. And this is just an example of a standard vaccine. If you're trying to mimic an attenuated vaccine, you have the protein, you would have a small molecule adjuvant, you can have a lipid in there, you can have DNA or some nucleic acid-based molecule, but loading all 4 of those together is a challenge. In this case, we can just throw all of them into the system. They will all load at about 90 to 95% efficiency, and it allows us to make customize the amounts of each one of these very nicely. It still releases inside of cells. They degrade inside of the endosomes, likely due to the differences in the chemistry inside of the endosomal compartments. And we can use this to have, say, very unique adjuvant effects and activation effects of these immune cells. This is just showing they're non-toxic in cells and have a standard biodistribution. They accumulate in the liver, depending on the size and structure. And shown here was just one of our optimized multi-component vaccine formulations, which is able to get a really nice antibody response after only about a week after the injection. Typically, an antibody response, a decent one, takes about 10 days. And so this is compared to a standard, just one adjuvant, one antigen formulation. Having this multi-component formulation actually has a lot of optimization. So we're working on high throughput analysis right now to optimize some of these systems for more rapid vaccine design. So those are just a few of the different systems I want to talk about. And it looks like I'm running a little low on time. But I wanted to give some examples of how we can apply these materials, particularly for the area of targeting strategies, which is a big focus for my group. So I'm going to transition now to how do we use these materials and the interaction at the nanobio interface for targeting. And I can briefly go through this. I'm sure everyone here is familiar with the fact that PEG, of course, is really important for coating on materials in order to decrease non-specific protein interactions and cellular interactions. And it was critical for the design of nanoparticles in the market, like Doxil, shown here, which took a really long time because of the issue with not having PEG on the surface resulted in rapid clearance. And so PEG decreased the clearance rates of these particles, allowing them to have an anti-tumor effect. But what's really an issue still is that even though you have that PEG coating, and even if you put targeting ligands on the surface of the particle, the mononuclear phagocyte system, the system of immune cells, essentially innate immune cells throughout our body, does not care. If you have PEG on there, it doesn't care if you have a targeting ligand. It is constantly sampling the fluid around it, and it will take in nanoparticulates. And that's still why to this day, even with the targeting moiety on the surface of your particle, you're still going to have the vast majority taken up in locations where these MPS cells are located. And they're all over the place. They're in the brain, they're in the lung, particularly the liver, of course, is the big area where they're going to be taking up these particles. So we want to incorporate not just methods of enhancing targeting, but also methods of trying to deal with these myeloid cells that are taking up our particles indiscriminately. And the interface, the nanobio interface turned out to be pretty critical for all of these. So I've broken these targeting strategies into 4 different areas. First is the classical one of active targeting. And our addition to this area is the fact that instead of just putting a targeting ligand on the surface, we do in vitro optimization of the interaction between the ligand and its target receptor. So many cells will express a target receptor at a certain amount, which also means that we can actually specify the ligand density on the particle to best match that, and also in many cases decrease uptake by other cells that might have a similar receptor but expressed at a different level. So we can do optimization in vitro to enhance the targeting, to further optimize our display of our particle targeting ligands. We do passive targeting. So this is without any kind of specific chemistry. It's just the fact that the shape of the particle inhibits uptake by certain cell types. And this is shown quite nicely by Dennis Disher many years ago. Filamentous particles were not taken up as well by certain immune cell populations. It has to do with the aspect ratio of the particle and the ability of endocytosis to fully engulf those particle types. So the shape is kind of a passive way of controlling targeting. Something that we recently published on, isn't used very often, is what I believe is a combination of active and passive. It's passive because we're not specifying a targeting ligand, but it's active because we are specifying the surface chemistry. So essentially what we're doing is that we're specifying the chemistry of the material to control the structure and composition of adsorbing proteins on the surface. And I have a few slides going into this in a little bit more detail. But essentially, we control the conformation and structure and content of the protein corona in order to specify interactions with immune cells. So that's one other way that we've started getting into. And finally, this is a newer idea, which I was actually kind of surprised that hadn't been tried before, but we're calling this an indirect targeting. Essentially, you can enhance targeting by decreasing the uptake in non-specific locations. So what that PEG corona was doing for Doxil, for example, was enhancing the circulation time, increasing the time it had to get to the tumor by decreasing the non-specific uptake. So if you actually were to, say, inhibit those cells that were taking up these particles indiscriminately, and then follow up with a second therapeutic particle, you could actually have much better targeting of that therapeutic particle because you've inhibited those initial cells. And so this is what we're calling indirect targeting. We designed a temporary non-toxic inhibitory particle to inhibit macropinocytosis specifically. And so typically, if you would inject a nanoparticle shown here, you will see accumulation in high levels in the spleen and liver, for example. But if you do a pre-injection of this inhibitory nanoparticle, you actually have drastically decreased uptake in those off-target locations because those cells are no longer able to undergo endocytosis. You can now have enhanced circulation of your therapeutic molecule or nanoparticle as a second injection. So we found this actually worked pretty well. And so this is actually just published or just accepted. I should have changed it. It just came out. It has a DOI now. It was available yesterday, I believe. So I'm going to go through quickly some of these different methods. So the passive targeting, the structure is quite important for this. We found that keeping the same surface chemistry but changing the actual self-assembled structure results in drastic differences in uptake at the organ and cell level. This passive targeting for us is pretty important for vaccines because it turns out that one structure, the vesicle, actually has a really nice predisposition to be taken up by dendritic cells. For a very long time, we didn't know why that was the case. And this latest paper we have actually demonstrates that this is actually due to the actual protein corona that forms on these vesicular structures that specifies that. So this is an example of using passive targeting to target dendritic cells. No targeting ligands, just a vesicle loaded with a very hydrophobic and toxic adjuvant, CL075 shown here, so a derivative of imimicrimod. We have a collaboration with Boston Children's Hospital, their Adjuvant Discovery Center, Precision Vaccines Program. They can send us a lot of adjuvants. We load them into our particles and send them back for in vivo and in vitro analysis. They did really nice human cell experiments for us. And so this shown here, we were able to make a vaccine, this one mimicking the BCG vaccine in terms of the immune response that you'll get, just by making a passively targeted particle that can enhance uptake by dendritic cells. This is just quickly, in neonates, they have very different immune responses than adults. And so targeting dendritic cells, neonatal dendritic cells with this adjuvant actually was very beneficial for making infant vaccine immune responses as good as adult responses. Shown here is another passive targeting strategy we did, also in the vaccine area, where we designed a nanoparticle to load lipids. So this is an example of a unique kind of antigen. It's not a peptide antigen, it's a lipid-based antigen. It actually results in activation of a completely different set of T cells that are lacking in many different subunit vaccine formulations because we don't put targeted antigenic lipids into our systems. We use lipids mostly for adjuvants, not as the actual antigenic target. So there's actually a really unique biology here where mice don't have these T cells where we do, and it makes it very difficult to study. So this collaborator here has made these humanized mice that are able to respond to lipids, lipid antigens, but she couldn't deliver the lipids because it's hard to deliver lipid payloads. So this is where the polymer chemistry came in. We made these nanoparticles optimized for loading this, it's called mycolic acid. It's a lipid that's part of the membrane, the cell wall of many mycobacterium. And we can now activate and study in a controlled fashion these different T cell populations that haven't been able to be studied before. And so in this case, we also looked at intranasal administration of our particles and such as well. Shown here also is passive targeting. Just by being able to get the drug into a cell of interest, you can enhance the efficacy of a drug that has intracellular activity. And so in this case, intracellular activity is really important if you have a parasite living inside of your cells. So simply enhancing the ability of a drug to get inside of a parasitic or parasite infected cell drastically enhances the efficacy. And so this is an example shown here. I'm not going to go into too much detail, but this is a parasite called a trypanosome. And we developed these nanoparticles that delivers a drug, benznidazole, that kills these parasites. The drug is fairly toxic, and so many times people have to stop treatment because of this toxic effect. We could actually decrease dosaging by about 600-fold simply because we could enhance the delivery of the drug into cells and selectively to the cells that typically hosted the parasite. And so this was done in vivo as well. This parasite actually results in heart disease, mainly the inflammation of the heart. It's one of the leading causes of heart failure in South America. And I hate to say this, but this bug is actually spreading due to climate change to the US. This was usually isolated to South America, and we actually found this bug in Chicago a few years ago. So I think we're being a little preemptive, hopefully having something, viable as a treatment that we're working on now, just in case in 10 years this could become an issue. But essentially, we can completely ameliorate this inflammation that you see in the heart in mouse models of these trypanosomal infections. So switching to targeted delivery. So that was passive, just using the shape. In this case, we needed something more selective. So we were interested in targeting inflammation in the heart. And in this case, we have a very complex situation where in the vessel walls, we actually have accumulation of lipids that results in atherosclerosis. But inside of these plaques that form is actually an immune microenvironment that's full of macrophages and small amounts of dendritic cells. But it turns out the stability is highly dependent on T cells inside of these plaques that are activated by the dendritic cells. So a challenge is, how do we target dendritic cells when we have a ton of phagocyte-like macrophages present? And so this is where we did a combination of the propensity of dendritic cells to take up the vesicle, along with a targeting ligand that we put on the surface of these particles. In this case, we're delivering something anti-inflammatory, vitamin D, in this case, to target the vitamin D receptor. It's really interesting. This goes into our repurposing, but vitamin D, it seems really basic, but it is extremely potent anti-inflammatory if you can enhance its delivery into cells and selectively into certain cell types. It's very good at inhibiting NF-kappa B through multiple pathways. And so simply loading something as common as vitamin D into these particles and enhancing the targeted delivery to dendritic cells. In this case, we used a targeting ligand shown here, this construct. This allows us to control very nicely the density of the targeting ligand on the cell surface by incorporating a lipid tail. So we have a lipid tail, peg spacer, and the targeting peptide. And we can really nicely control the density of this targeting peptide because the amphiphile loads so nicely inside of our self-assembled systems. And this allows us to do in vitro optimization studies. So we can say, for example, optimize the targeting of a receptor that's present on macrophages and dendritic cells, but express higher on the dendritic cells by controlling the display of this targeting ligand shown here. So this is in vitro optimization, showing there's a sweet spot with a specific peg length, as well as surface density that results in optimal uptake by the dendritic cells and decreased uptake by the macrophages. And so we did this in vitro first, and then we tested it in vivo. And just quickly, we were able to very nicely enhance uptake by dendritic cells within atheromas, within the aorta, without impacting this background signal that you get from uptake by macrophages. And I'm going to be honest, this background signal from macrophages is you just really can't avoid that. We're hoping that our new indirect method could enhance this, but we haven't tested that yet. But yeah, you're going to have background macrophage uptake. But the question was, can we enhance uptake by the cells of interest? In this case, dendritic cells, we could significantly enhance their uptake within plaques and the spleen for these anti-inflammatory treatments. This is just showing how anti-inflammatory treatment of dendritic cells works with vitamin D. We're decreasing, as I mentioned, co-receptor expression. We're decreasing MHC2 expression, which is correlated with these inflammatory cell types. We're decreasing inflammatory cytokine expression like IL-12 and enhancing tolerogenic expression of cytokines like IL-10. And the result is a really nice activation of regulatory T cells. I think I'm running out of time, so I'll just quickly go through just showing we decreased plaques. We quantify this. We have decreased inflammatory cells within the vessel wall, as well as decreased lipid accumulation. And it was at a very low dosage as well. And so we're actually investigating now using a slow release system, our hydrogel system, to slowly release vitamin D over time to maintain high levels of regulatory T cells in many different situations, which we find to be quite I thought I had that slide. I must have taken that slide out, but that was actually quite interesting because we can actually increase regulatory T cell levels generally in a patient or in a mouse for long periods of time. So we're investigating that treatment for heart disease now. This targeting strategy works for many other situations. Shown here is recent work. We have a few papers now studying glaucoma. So in this case, we're delivering molecules or a peptide that actually targets lymphatic receptors that are present in certain cell types inside the eye. We're interested in the Slem's canal, which has lymphatic characteristics, and so enhanced targeting of those cells, decreased targeting of other epithelium and endothelium within the eye to decrease side effects. And this actually worked quite well. We're working with Dan Stamer, who's a really big guy in glaucoma at Duke. He's doing a lot of these mouse models for us, and we can drastically decrease intraocular pressure in mouse models. So we're investigating now different ways of controllably delivering these particles. And the last thing I want to talk about, which I found really interesting, is this control over the interface at the protein nanoparticle interface. So shown here is just the accumulation of proteins on the surface of nanoparticles. And what's really interesting about this is that this is one of the innate forms of the immune system, is that when proteins stick to nanoparticles, they denature. And that allows them to interact with scavenger receptors on the surface of these immune cells, which again, they just take up, that's their job. They're supposed to take up things that shouldn't be there. So by controlling the denaturation of proteins, you can actually have a mechanism to control uptake by these cells. And that goes down to the chemistry of your nanomaterial, because that dictates the structure that forms in terms of how the protein unravels. And so shown here is multiple chemistries that we tested. This is a control that's going to be complement activating. We have one that's standard. This is the most commonly used chemistry in vivo in the clinic right now, methoxy. And this is something that mimics the chemistry you see on the surface of cells, so phosphate groups. And so we want to investigate, if we change the chemistry here, how does that impact the types of proteins that stick in their conformation? It turned out that this had a huge impact on the biodistribution. So same particle, same size, only thing changed is that surface chemistry. and the phosphate group actually result in the most significant differences from the other two groups. For example, if you want to avoid the kidney, it actually turned out that certain myeloid cell populations in the kidney just did not take up these particles, as well as the same in the lung. We saw differences. I don't have time to go through all through it, but many different myeloid cell populations differed between the uptake in those three different surface chemistries. So what was going on here? So first we looked at what was the major protein absorbing. Albumin, which is kind of obvious, turned out to be the main component. But we actually found something fairly unique about this situation, is that so first, the enzymatic accessibility for degradation of albumin differed on the phosphate surface versus the others. So it actually was maintaining a more natural conformation when bound. It wasn't denaturing as much. And this was backed up by circular dichorism. And it turned out that albumin was actually denaturing into a state that enhanced interaction with scavenger receptors, particularly this one, SRA1. And so we use a variety of different types of inhibitory platforms to study, is it this receptor? What receptor is it? It turned out to be SRA1. And so by specifying the chemistry at the interface, we could control the conformation of BSA, which actually had a huge impact on interaction with scavenger receptors on these cells. And so we're actually looking into this quite a bit more. We just finished a study where we did a comprehensive proteomic analysis of all the proteins that are sticking in human blood to many different cell types, and how this actually, different nanoparticle types, and how the structure actually impacted that. It turned out that not just the chemistry, but the combination of the chemistry and the structure had a huge impact. So we looked at what were the similar proteins sticking in all these different cases. And this is actually the impact it had on cell uptake within immune cells. There were actually combinations of surface chemistry and structure that had either no uptake versus super high uptake versus different nanoparticle types. And so we're actually using this right now to try and find out, is there, say, characteristic proteins within the corona that we can use as markers to predict uptake. So we're trying to use this to predict what kind of interactions you'll have in a patient or during a delivery situation, depending on the combination of chemistry and structure of the materials. And so this is in revision now. We hope the comments were minor, so we hope to have this out pretty soon. I think I'm almost out of time. So I did want to talk about this indirect targeting. So essentially, I did kind of describe how this works. Essentially, we have an inhibitor that is able to inhibit uptake non-indiscriminately by macropinocytosis of these cells. So shown here is we have chlorpromazine, which is going to inhibit receptor-mediated, whereas letronculin, shown here, actually inhibits macropinocytosis in this case. And so we can show that we can actually have receptor interactions we can have receptor interactions while inhibiting endocytosis via macropinocytosis. Letronculin is a little toxic, but in the nanoparticle form, it was not. And so this is how this strategy, I do want to stop here after I get through this. This is how this strategy actually works. So say you have a tumor model, you can inject with this inhibitory particle first. It's going to distribute where nanoparticles normally go, and it's going to bind or be taken up by those cells that indiscriminately take up our particles. And if it has an inhibitor in there, we can temporarily, for say a day, 12 hours, depending on how much you deliver, inhibit those cells from undergoing endocytosis, resulting in the ability of a second injection to now circulate longer and get to the target location. We spent quite a bit of time optimizing the schedule for this. It turned out that a pre-injection 24 hours ahead of time, and then a co-injection of the effector, we're calling it the effector, the therapeutic drug, along with the inhibitory nanoparticle. This was the most effective, this one right here. And so you'll see decreases in the spleen and the liver. You have enhanced delivery of the nanoparticle to blood following a sub-Q injection, which was interesting to us, showing that we're inhibiting uptake within lymph nodes. And it had an up to 8-fold enhancement in targeting of immune cells and tumor cells inside of the tumor microenvironment. So it actually was able to enhance a target. This is just a generic folate targeting strategy. It was able to enhance drastically, significantly, receptor-mediated uptake within the tumor by inhibiting non-specific uptake elsewhere. So we're actually submitting some grants right now to further study how we can enhance this or use this in other areas, and further study how this can be designed and optimized for controlled delivery applications. So I'm going to stop here. This just goes over repurposing of drugs. We repurposed rapamycin to have a completely separate mechanism of immunosuppression, but this is something I can talk to Daryl about later. He'd be more interested probably in this enhanced blockade that we're seeing, co-stimulation blockade that we're seeing. So I'll stop there for the sake of time, so we have questions at the end. I just hope I was able to give an overview of the applications and design of our PEG PPS systems, the two different forms, the homopolymer system and the blockopolymer system. And these are just some of the many different applications we're working on now. Try to give a few examples of those that we published on already. And I'll stop there. These are our funding sources. And this is, of course, the group that did all of this work that I described today. So I'll stop and I'll be happy to take any questions.
Great, thank you, Evan. Hope everybody can join me, use your reactions to say thanks for a great seminar. If you have questions, if you can raise your hand in the participants window, I'll call on you, or you could put your questions in the chat. I hope folks can hang out for a few minutes to ask some questions. Maybe I'll start. So the ppsu polymer behavior was really fascinating, and there are a bunch of questions I can think of there. The first one I was wondering is, are these kinetically trapped and stable structures? Like if you form a particle, do they remain in that initial particle state for a month, or do they, over time evolve to something else, or what do you know about?
Yeah, very good question. It turns out they are hyper stable, extremely stable. They were actually so stable at first that I was worried that we wouldn't actually have any intracellular delivery, but it does turn out that the endosome is able to degrade them. But we have them sitting on the shelf for, I guess after this study, like a year now. No change in size or structure. They're actually extremely stable. And the payload stays inside during that time as well.
And then what's the mechanism by which they break down eventually? Do they just slowly dissolve, or can those polymers be proteolytically cleaved somehow?
The only way we're actually seeing degradation in an in vitro setting is by shifting the polarity of the solvent again, which disrupts the sulfone interact. Anything that can disrupt the sulfone interactions will result in degradation. And so we believe that that's what's going on inside the endosomes. We have at least a harsh enough chemistry for that to occur. But if you keep this in PBS on the shelf, this just doesn't happen. And so as far as we can tell, shifting the polarity of the solvent is the only way to cause them to degrade.
If there aren't other folks from the audience, I'll ask one more. So the inhibitory particle strategy seems really interesting. So is the action of the initial particle carrying the latrunculin, is that a reversible process? So they're inhibited for some period and then eventually they would recover their phagocytic activity?
Yes, exactly. So latrunculin is actually an inhibitor of actin polymerization, but it's temporary. And so depending on how much you give to a cell, you can inhibit, or essentially you break down its cytoskeleton temporarily. And so our collaborator had been working with this. So it's kind of funny. This actually started with our glaucoma work. Sorry, I have two screens and it's shifting back and forth for some reason. This actually started with our glaucoma work. So our collaborator had actually looked at using latrunculin to soften cells within the eye to allow increased fluid flow for exiting the eye. And so when I saw the video of them doing this, it shows the actual disruption of the cytoskeleton in real time. You can actually see them break apart if you have a fluorophore bound to actin. We were using latrunculin at the same time to just look at, can we actually inhibit, endocytosis? We weren't thinking about it so much in vivo at that time. But when we saw that it could actually disrupt the cytoskeleton so quickly, but then it recovered over time, because you could see that with the microscopy, then we were like, wow, this is actually temporary. And if you give it a low enough concentration, it was non-toxic. So that was actually part of the getting that schedule to work out in terms of the injection schedule, figuring out how long ahead of time we should inject, how much we can inject to make sure it's within that window of time frame where you still have an effect that's not toxic. And so it turned out within about 24 hours, that effect pretty much goes away. And we did a few studies to show that we could inject, 2 days later, and you would have normal biodistribution of the particles. So it was completely reversible, just based off of the time. I was actually kind of surprised this hadn't been tried before, because people had used clogenate nanoparticles to deplete macrophages and such. So people thought about killing the cells and destroying them, but not temporarily inhibiting their uptake. So I was kind of surprised it hadn't been really tried before, but it seems to work.
Thanks, everyone, for tuning in, and thank you, Evan, again.